starting chapter 12. Okay. So, first thing is the idea of communication. Understand there's two major communication systems in the body. We have the nervous system, which is the focus of this test, and the endocrine system, which is going to come up in a few tests after this. Have an idea, compare the two. Okay, just have an idea of the comparison between them. Okay. We said functions of the nervous system. We can also integrate this idea into the, we talk about the, okay, so chapter 12. So, two types of communication in the body, right? One is the nervous system, one is the endocrine system. Endocrine system is another test, okay? But you should have an idea of the comparison of the two. Okay. Now, the nervous system has these functions. Don't get these confused with the divisions of the nervous system. There is some overlap here, but these are the functions, not the divisions of the nervous system. So at its base, we have a sensory function. Detects changes from the outside, conveys them to the inside. You should know that these are carried on. Sensory neurons are afferent neurons, and I emphasize the on this, okay? Afferent neurons. But in class, I don't pronunciate the word exactly right because I'm trying to give emphasis on the A, okay? But you should know what afferent neurons are and that they carry information from the outside to an interneuron and then to the central nervous system. Another function is this integrative function where we take signals from both the outside and from the brain and move them around. And we talked about this when we talked about the divisions of the brain, when we talked about tracks and moving things around. All goes back to this idea of the integrative function, where we take different pieces of information and integrate those and come to an overall decision and communicate that out. Okay. Finally, we have the motor function. These are carried on efferent. I emphasize the E here, efferent neurons. Okay, these affect a change, and you should know the difference between afferent and efferent. And again, I'm overemphasizing the E to make the point for you. You should know the types of nervous tissue. The nervous system is made of two types of tissue. We have neurons and neuroglial or glial cells. And have an idea of what each one is doing. Okay? But there are two main types of cells in the nervous system. For the test, you should be able to label these diagrams. If I give you this on the test, I would give you a diagram that is in the lecture slides. Okay, I will not give you a diagram that you have not seen before. Okay, so I'm going to go through these. The cell body, this has the machinery, because ultimately this thing is a cell, right? And you go back to intro biology and you have those generic pictures of cells. Well, that's true, but again, you know, a neuron is still a cell, has a nucleus, has all these things. It's just a very specialized form of a cell. Okay, so it has a cell body, has the machinery. You know, because a eukaryotic cell is going to have a nucleus, right? It's going to have a way to make protein. It's going to have a way to do oxidative metabolism. It's going to have that mitochondria. It's going to have all the machinery of a cell because, indeed, it is a cell. And that machinery, if you will, is located in the cell body. Okay. Dendrites, another important feature here. These are going to receive the information from the outside. This is where the input of information happens. The axon, we talked a lot about this. This is going to carry that information away from the cell body to the end of the axon, at least in the multipolar axons. We talked about different axons later on, so be aware that the picture we have on the right here, this is that multipolar type. This is what you're going to usually see when you see a picture of a neuron, but this is not the only shape to it. Okay. The hillock, trigger zone, know what those are. Again, that's where it connects to the cell body, and that trigger zone is where that initial threshold voltage is going to be important. Okay, that's that. Think of that as that first domino that clicks down and starts the process going. Synapse, another very important area. This is the end of the axon, and this is where neurotransmitters are going to release from. It's also where reuptake will happen. Talked about types of synapses, chemical and electrical. The one you see here with neurotransmitters, that is a chemical synapse. There is electrical synapses, not nearly as common as your chemical synapse. For every one electrical synapse, you're going to have like 99% plus chemical synapses, but they do exist. Synaptic vesicles, those are balls, if you will, of neurotransmitter that are made in the cell body where the machinery is, and it carries it down to the end of the axon, okay, to that synaptic bulb where it's released. The cleft, another important thing, that's the area between the synapse and the postsynaptic cell. This can be a muscle, okay? This could go to a gland. This can be any number of things, but there is a region after the synaptic bulb. Neurotransmitter, we talked a lot about these toward the end of the material for the test. This is what is released, and then the postsynaptic cell is what it releases onto. Remember the neurotransmitter, this can be, for example, could be a ligand. Okay, this can cause a ligand-gated channel to open up, let ions in the case of muscles. It also can sit on something and start a cyclic AMP pathway. We didn't talk too much about that quite yet, but it doesn't have to let anything in. It can actually change something internally in the cell. And we'll talk more about that as the semester progresses. Okay, and also here, very important is to know the information flow on a neuron. Okay, it's going to go through the dendrites, cell body, and then pass along the axon. It does not go axon, cell body, dendrites. Okay, there's only one flow of information down this. Axial transport. The axon, okay, is an empty tube, essentially. Right? Now, empty as in it's filled with fluid, you know, it's not like an empty PVC tube. Think of it as maybe a PVC pipe filled with fluid, right? It has to have some fluid in there. It's not like a hollow space. But that area is essentially a hollow tube filled with fluid. Important is that we're going to move stuff down the axon and up the axon. Okay, we're going to move stuff from the cell body down and stuff from the end of the axon toward the cell body. Really important to understand the physiology of the neuron here, because the cell body is where the machinery of the cell is located to make things like neurotransmitters. Okay? The end of the axon is simply not suited for that function. Okay? It's specialized for the conduction of the action potential, not to make those neurotransmitters. So the cell body is where the neurotransmitters are made, and axial transport carries those down to the end. Okay. And that's this point here, is that, you know, substances are made in the cell body, the neurotransmitters are made in the cell body, and they have to be carried down toward the end where they can be used. Now we said there's two types, antegrade and retrograde. Know both of these. Antegrade is from the cell body to the end of the axon. Retrograde is from the end of the synapse to the cell body. Also know that this idea of retrograde, you'll touch on this in microbiology, right? We talked about a good example of rabies. It's going to enter through the end of the synapse and progress toward the cell body and then toward the brain. Okay, this is why it takes, you know, several weeks to show symptoms of rabies from an initial infection, because it actually has to progress up the axon via retrograde transport. Classification of neurons, structural classification. structure, you should have an idea of all three structures. I could give you one of these pictures and say, what is this? So have an idea of these three shapes, okay, and what they do. If I gave a picture of these, again, I would give these pictures in the PowerPoint. Okay, so that was a structural classification of neurons, right? We got multipolar, we got bipolar, unipolar, or pseudo-unipolar, whatever you want to call it. But there's also the idea of a functional classification, right? Based on what they do, not their shape. And we covered this, a few minutes ago, right? That we have some neurons, the afferent neurons, or I like to say afferent to emphasize the A, that are going to carry information to the central nervous system. These are affected by the external environment. Motor neurons are efferent, emphasis on the E here. These are going to carry information from the brain out. These affect a change. Okay? Another way you might remember it is think of them alphabetically, right? A before E, you get hit on the hand. I just hit myself on the hand. An afferent neuron detected that, takes it to the brain, say, hey, you just felt pain. And then it moves that information to another neuron, to a muscle, which is the efferent or motor nerve, or neuron. Okay? So know the difference between the two. Interneuron, we talked about this with the reflex pathway. This is going to connect neurons to one another. We'll talk more about that. We talk more about this later in the class. Okay, what we have gone over, essentially from the start of this, we said what? There's two types of nervous tissue, right? We have neurons, and then we have this glial or neuroglial type. What I've talked about up until this point is mostly that neuron type of tissue. Now we're going to talk about the glial or neuroglial cells. Okay, this is the second type of cell that makes up the nervous tissue. Again, know the two. Neurons and glial or neuroglial cells. These are the supporting cells that keep everything going. Okay, so what do I mean by that? Neurons, okay, they are specialized for the conduction of information. Okay, but because they're so specialized, a lot of other things have kind of gone by the side so they can focus on their function of conveying information. So we've got to have these support personnel, if you will, or the glial cells that take care of the basal activities, that take care of the housekeeping activities. Okay, that goes to protection. Okay, we'll talk more about that. That goes to structurally securing the things. Okay, that is the idea here, that the neurons are specialized for one function, but because they're so specialized, a lot of other functions, they don't do very well or they don't do at all. So they have to have the glial cells to support them. Now we talked about UAB, the Center for Glial Biology, not on the test, just more of an FYI for you. We talked about what? There are different glial cells in the central nervous system and the peripheral nervous system. You should have an idea of the function of each and their comparative one in the peripheral versus the central nervous system. Okay, a little bit more on that in a minute. But also, see the ependymal cells, those are important when we talk about the what? Cerebrospinal fluid. So let's see that in a minute. Okay, so the first type are the astrocytes. Okay, several functions for astrocytes. I'll let you read through these. Oligodendrocytes. These are going to make the myelin sheath in the central nervous system. Very important. You should also know their comparative type in the peripheral nervous system, which are the Schwann cells. Okay. Does the same function, just different regions. Central nervous system is oligodendrocytes, and then the Schwann cells will do the same function in the peripheral nervous system. Microglial, these are going to be more for protective things and to clean up waste from around the neuron. Again, they're so specialized in their function that they don't have this ability, right? They have to have these supporting cells to do this for them. Ependymal cells, these are important in the production of cerebrospinal fluid. We will talk about these in the next chapter. Neuroglial of the peripheral nervous system. Schwann cells are the most important ones here. These are going to form the myelin sheath around the axons in the peripheral nervous system. Okay, the oligodendrocyte is their comparative one in the central nervous system. Okay, myelin sheath. Myelin sheath, the idea here is that, and we talked about this later on, remember we'll talk about this toward the end, is the idea of myelination. That going down an axon, there are regions where we don't have the movement of ions, right? And that's important because that allows the speeding up of the action potential changing along the axon. And we'll talk more about that later on. But the myelin sheath is a lipid layer around the axon which prevents the conduction of ions across it, and by separating regions where conductivity happens, we speed up the signal. And again, we'll talk more about that when we talk about action potential. We call this, incidentally, saltatory conduction. Node of Ranvier, very important. These are bald spots, or gaps between that myelination. Okay, do you see how we have on the bottom picture we have the blue jelly bean looking things, right? Okay, that is myelin sheath. The areas of no myelin sheath in between these, those are the nodes of Ranvier. And that is where ions move, and we have that action potential. Okay. Again, know what the node of Ranvier is, and know its function. Gray matter versus white matter. Gray matter, well, let me start with this. First of all, you should know that there's different locations for gray matter and white matter depending if it's in the brain or the spinal cord. Okay. On the left, you see how we have the light tan material on the outside of part A. Okay, that is the white matter. In the interior, we have gray matter. Look on panel B. We have the reverse. On the exterior, we have gray matter, and on the interior, we have white matter. Now, there's some regions of gray matter in the interior, but for the most part, in the interior of the brain, we have white matter. Now, gray matter lacks myelination. Okay, the white matter has myelination. Okay, the next topic, major topic, is the movement of a signal down a neuron. Okay, we're trying to get the idea across of how you get a signal, say, from the interneuron in your back all the way to a muscle in your finger. How does that happen? That's the core idea here. Now, before we start, I want to bring something to your attention about a pronunciation of a word. The node of Ranvier. Now, I was taught it was pronounced Ranvier. There's lots of different pronunciations for this. He was a French physician. Some people are going to call it Ranvier. Some people would call it Ranvier. You're going to hear different pronunciations for this. Typically, Ranvier tends to be the more accepted or the more common pronunciation. Okay, so just be aware that if you hear a slightly different pronunciation, he's a French physician and it's a French last name. Different people are going to pronounce it slightly differently. Okay, so let's begin. Now, just remember, we are talking about the movement of a signal along the neuron. That's the core idea we're talking about here and what you have to know for the test. Permeability, this is the word we use to describe how easily an ion can move from one side of the membrane to the other side. Going back to membrane physiology, recall that what? The membrane is what? Bipolar, right? There are polar parts on the outside, and on the inner part we have that nonpolar region, and ions do not easily pass across that. There has to be a channel, a tunnel, to allow those to pass. Okay? So in our discussion about what's happening at the node of Ranvier, we will say it's permeable to this, not permeable to that. What we're really saying is how easily the ions can pass from one side of the membrane to the other side of the membrane. That's what we mean when we say permeable. Okay? Know what that is. Also, that ions have to have a channel to pass through, right? And the opening and closing of this channel is usually a regulated event. Okay, meaning that there has to be a specific voltage, a specific ligand present to open or close one of these gates. Not always the case, but for what we're going to discuss here, we're just going to look at those ligand-gated channels, okay, and voltage-gated channels. So you should have an idea of what an ion channel is and why it's needed. What is a channel? It's a pathway to allow ions from one side to the other side. What's the differences? Well, why do you need it? Because ions don't go from one side to the other side without a channel or pathway for them. And then third point, and very important, is different things are going to open and close these channels. Okay? Some cases there's nothing keeping this open or closed. A leak channel will randomly open and close. Okay? Another type of these channels, ligand or ligand-gated channels, something has to sit on the outside, a neurotransmitter for example, and that will open or close these channels. Another type is mechanically gated channels. The movement of the membrane around it will open and close the door and allow certain ions through. And then the fourth and probably the most important for this discussion is the idea of a voltage-gated channel. Okay? Voltage-gated meaning that that door that allows ions to pass will open and close at specific voltages. Okay? There's going to be different ones for sodium, there's going to be different ones for potassium. The voltage that a sodium gate opens is different than a potassium. Okay? So the idea is this, core idea. 1. The movement of ions is important in the conduction of a signal down a neuron. Key point. Second point is these ions that move in and out of the neuron move via ion channels. 3. The ion channel opening and closing is a regulated event. The regulation we're going to look at today is the voltage-gated channels opening and closing. Okay, that is the key one for this discussion. Okay, so we talked about the different channels and just have an idea of which ones they are. Leak channels, and again, this allows specific ions in and out. There's a leak channel for sodium, there's a leak channel for potassium, but they don't work interchangeably. There's a ligand or ligand-gated channel. Okay, a neurotransmitter will sit on this channel and cause that channel to open up and allow a specific ion inside or outside. Probably saw this in muscle physiology. What starts a muscle to contract? Mechanical gated ones. You should know what this is. Mechanical stimulation opens and closes these, important in touch reception. And the most important one for this discussion is the idea of a voltage-gated channel. These open and close at specific voltages or potential differences to allow certain ions to pass. There's a potassium, there's a sodium, and they both will open and close at different voltages. Okay, so when we talk about what's happening at a node of Ranvier, we will probably say at this voltage, sodium or potassium comes in or out. Why is it coming in or out? Because a voltage-gated channel is opening or closing. Why is it opening or closing? It's because that voltage that it functions at has been reached. Okay, so when we talk about the movement of ions and the change in voltage, bring it back to the idea that that's opening and or closing a voltage-gated channel. Okay. Where are you going to find the movement of ions? Another important concept. You're going to find those primarily at the node of Ranvier. Okay. That's where you're going to find it. And we're looking at the process on a cellular level as what's happening to get the signal down this axon. Very important. When we talk about this, we're talking about what's happening at the node of Ranvier. We're not talking what's happening underneath the myelination. Okay? We're not talking about what's happening underneath the myelin sheath. We're talking about what's happening at those bald spots, at those nodes. So what does a neuron at rest mean? And we say that a lot, a neuron at rest. You should know what a neuron at rest is. It's like a coiled spring. It's like a series of dominoes stuck in a row. It's like people in a stadium all sitting in their seats, getting ready to stand up to do the wave. Okay? This thing is charged, ready to go. Start at one end, and the movement of the different parts will communicate that information to the other end. I like to use the analogy of dominoes, right? I have a series of dominoes that are about 1 meter long. When I push one domino, they consecutively knock over the next domino, right? And we get to the end of the dominoes, and that last domino falls down. Okay. When a neuron's at rest, think of all those dominoes sitting up, ready to go. What are the ions inside and outside? You should know the 4 major ions. Of particular note, be aware of potassium and sodium. Okay, potassium and sodium. What are the concentrations of the ions inside and outside? Negatively charged proteins, very high on the inside, non-existent to low on the outside. Potassium, very important you know this. It's very high concentration on the inside. Sodium, very high concentration on the outside. Going back to chemistry, think of what's going to happen with the concentration gradient. What direction are these ions compelled to move because of their concentrations? Okay. You should be able to draw this picture on a test. This is a node of Ranvier at rest. This thing is like a charged coil ready to go. Okay. And again, you should memorize these concentrations. Of particular note is the sodium and the potassium. How are they going to move? They're going to move through voltage-gated channels, in some cases leak channels, but we really are going to talk more about the voltage-gated channels. They move through channels. What do we mean by polarized, right? We say it's charged, and I've said charged a lot. What do I mean by that? I mean that the interior is more negative than the exterior for a resting neuron. When there is a difference in charge across the membrane, where the inside and outside is of different charge, that is polarized. Okay, there's a difference in charge. If we can measure the charge on the inside and measure the charge on the outside, we would see those two numbers are different. When those two numbers are different, it is polarized. When those two numbers are the same, it's not polarized. Okay? The membrane at rest will have more negative charge on the inside than on the outside. We give a number that of minus 70 millivolts. Different number for different neurons. There's a little bit of fluctuation with that number, but at rest it's approximately minus 70 millivolts. Okay. What's the charge inside? At rest it's more negative inside than outside, and that's because the presence of ions and negatively charged proteins on the inside, the sum total of those make it more negative on the inside than the outside. How do we measure the charge? You should know when we say charge, we are really referring to the interior of the axon. Okay? We are referring to the interior of the axon. When I say it's minus 70 millivolts, or it is positive 30, or positive 35, or whatever number I mention, I'm assuming you know that I am referring to the voltage or the charge on the inside, not the outside. Okay, the number we always quote or always say is relative to the inside. Okay. And this is what it would mean if it's positive, negative, or 0. Okay. Okay. Very important you know this number. The resting membrane potential is minus 70 millivolts, meaning the interior is more negative than the exterior of the axon. What forces are acting on the ions? One you already know, right? If you've taken chemistry, you know there's a concentration force, right? Where if we have more concentrated ions on one side than the outside, on one side than the other side, that what? There's a drive for equilibrium. So if it's on a more concentrated side, it will move toward the least concentrated side. Okay, you're always going to be moving down the concentration gradient, trying to reach equilibrium. Okay. But there's also an electrical force on these ions, right? If it's negative or positive inside, that is important because those ions also have a charge. Do you think they're going to want to go to the opposite charge or the same charge? Well, they're going to want to go toward the side which is the opposing charge. Okay? Opposites attract. Right? So if we have a positive ion, right, it's going to be pushed or drawn toward the negative side and vice versa. That is, basic thing of just magnets, right? Opposites attract, likes repel. Okay. Okay, so we have a competing interaction of force here. A competing interaction of force. One's a concentration gradient, one is that electrical force. Sometimes those are both acting in the same direction. Other times those are in opposition to one another. Okay? Sometimes an ion will be more concentrated on one side than the other side, and it will be going toward its opposing charge. Other times those two forces are in competition. Think of it as like a headwind, right? That the concentration wants to take it from one side to the other side, but then the electrical force on it will be pushing against it and want to compel it to the opposing side. Okay, so you have to consider both forces on the ion. Okay. Okay. Action potential. Action potential is, and this is a good example of those forces acting on a particular ion. Okay, so you can use this just to see those different forces acting. So we got a chlorine ion, right? Okay, the green arrow is saying that chlorine right? Chlorine is a negative ion. Okay, so let's look at the ones on the left. On the left, it's 0 millivolts, and on the right, it's minus 65 millivolts. Okay, actually, voltage has a thing with distance, so this is not exactly a good image of it, but I'm trying to make the point of negative and positive here, okay, without getting too much into the physics of what voltage is. Okay, let's say those blue spheres are chlorine, right? On the left, we have 5 blue spheres. On the right, we have 2 blue spheres, meaning that we have a higher concentration of chlorine on the left than we do on the right. Okay, so if we just looked at it based on the concentration force, or the chemical force, the chlorine would want to go from left to right, shown by that green arrow. But what charges chlorine? Chlorine is a negative ion. Do you think it wants to go toward the side that is more negatively charged? No, it doesn't. There's an opposition there. There's an opposition of force, right? One force, the chemical force, if you will, wants it to go from the left to the right. But the electrical force, opposing charges, right? We have a negative going toward a more negatively charged side. That's giving a headwind toward it. That's giving the opposite charge. Which one wins out? It just depends on the concentration and the magnitude of the charge on the opposing side. Okay. So what's an action potential? Action potential is, and for the test, if I had a diagram like this, I would expect you to say, draw these arrows in, right? That is an expectation I'd have on a test. Okay, that if I set the situation up and I said these are chlorine and the it's more concentrated over here on the left and it's less concentrated on the right, I would expect you to be able to have an idea of what direction those forces are. Okay. Action potential, you know what action potential is, and this idea that when we look at action potential, we map it out on this 2-axis chart. The x-axis, okay, that will represent time. And time is not a lot of time. This is milliseconds. Okay. The y-axis will represent the internal charge in millivolts, or the membrane potential in millivolts. Okay. You should have an idea of what's happening at each point along this diagram. So what's happening when we have that initial stimulus, the depolarizing phase, and the repolarizing phase. Okay, now this is a nice just review of the concentrations, and the image below kind of gives you an idea of what's happening at each of the voltages. At minus 55, what happens? At minus 55, we have sodium-gated channels open up. Okay, let's go back to this and see what the concentration of sodium is. What's the concentration? High on the outside, low on the inside. Okay, so what direction will sodium want to travel in based on its chemical concentration? Well, it's going to want to go from the outside to the inside. So those channels open up and allow these positive ions to go to the inside. But let's think of the other charge here, right? Or the other force, that electrical force that I discussed before. Is the interior more negatively charged than the exterior? Answer to that is yes, it is. So there's going to be two forces acting on the sodium on the outside. The concentration force, if you will, which pushes it from the outside to the inside. And also the electrical force, because it's more negatively charged on the inside than the outside. Both of those are going to be very strong impetuses to push the positive ions from the outside to the inside. Why can those move? Because the membrane has reached minus 55 millivolts. And that's that key voltage that opens the door to those voltage-gated channels and allows the movement to happen. Why could it not happen before? Because ions being charged do not easily pass through the membrane. Okay? So, what happens? We reach minus 55. Voltage-gated sodium channels open up. Sodium moves from the outside to the inside. Why? Two reasons. Because one, it's more concentrated on the outside than the inside, and it is more negatively charged on the inside. It's going toward the side that is of the opposing charge, if you will. Maybe not the best way to say it, but it gives you the idea that the sodium is attracted to the side that is more negative because sodium is a positive species. Okay? So it goes out of there like a bat out of heck. Okay? And what happens to the interior voltage? So let's follow that green line going up. What's going to happen? We have a large mass number of positively charged species going from the outside to the inside. What's going to happen to the charge on the inside? It's going to become what? Less and less negative. It's going to go towards zero, and then at some point it's going to go and become slightly positive, more positive on the inside than on the outside. So as you move those positive ions from the outside to the inside will become more positive relative to the outside charge. Okay, and we see how the green line goes up and goes above zero and then goes to the positive side? That is why, because we're moving positive ions from the outside to the inside. But what happens once the interior starts becoming more positive than the exterior? Well, sodium is positive, so it is going to start hitting that. Remember we said there's that opposing charge where sometimes it can have a concentration and a charge force, and those can be in opposition. That's going to happen here. As it goes from the charge changes, you're going to push positive ions from the outside toward a more positively charged region. And there's going to be an opposing force there, right? Because you are going toward it's like putting two magnets together where you push both two positive poles on one another. There's an opposition there. Okay? Remember, likes repel each other, opposites attract. Well, you're getting positive ions going toward a side that is more positive than the other side. And that will cause a force of repulsion. There will be a repulsion there. Now, the other question you hear is, well, does this mean, how about the sodium? Will all the sodium just go through? Well, the best analogy there that I've heard is it's like taking a handful of sand out of the beach. Okay, you're not going to run out of sodium on the exterior. It's like pinching sodium. It's like pinching or getting a handful of salt or sand at the beach compared to the entire beach. You're not going to run out of sodium. Okay. So, know the depolarizing phase, right? Know that magic threshold, that what's happening, and then be able to describe the direction that sodium is moving and the forces on sodium as it moves. Okay, this kind of describes that. We have a chemical force and an electrical force, right? And I said the chemical force is the concentration, right? And then the electrical, that's the idea that the interior and the exterior of one will be more negative than the other, right? And you have the charge of an ion will be attracted toward the side that is of opposing charge. So initially, that positive sodium is attracted to the side that is more negatively charged. It's repulsed by the side that is more positively charged. Initially, that sodium will, when at minus 55, it will have a force on it to push it from the exterior of the node of Ranvier to the interior because of the chemical and electrical force. But as the interior becomes more positively charged than the exterior, that electrical force, instead of being a drawing force, is actually a slightly repulsive force. But the chemical force is so great that it overwhelms the electrical force toward the end. And then this kind of gives an idea of what's happening. At +1, what's happening, the chemical force is still pushing the sodium into the cell, right, into the axon. But the electrical force, we have a positive species going toward a more positive side, and that's providing a headwind, if you will. But overall, that electrical headwind is minor compared to the chemical force, which is greater. We start getting up to +10, and that headwind, that electrical headwind, if you will, is growing greater. But still, the chemical force is still greater than the electrical, and it is the overall net force is pushing sodium still into the axon. Okay. Now, at +25, +30-ish, in that region, something important happens, right? It's hitting a headwind, if you will, of the interior being more positive than the exterior. And that's slowing it down. Okay, that's slowing down the movement. At +30 millivolts, some things happen. One, those sodium channels close. The door that allows sodium to enter closes. Now we have potassium. Potassium is more concentrated on the inside than the outside, right? Going back to this right here, we see that potassium, K, is very concentrated on the inside, less concentrated on the outside. Okay. So just kind of remember that for this. Now, because of that, the interior is more positively charged than the exterior. So think of potassium, right? We have a positive species that's more concentrated on the inside than the outside. What direction does it want to go? Well, it wants to go from the inside to the outside. Think about the electrical force on it. Okay, the electrical situation. Is it positive or negative on the inside relative to the outside? Well, it's more positive on the inside. Okay, so both of these factors will favor the movement of potassium from the inside to the outside. Now think of it this way. Potassium has a positive charge. If we somehow have a mass exodus of potassium, what happens? The interior is going to lose a lot of positive charge, and the exterior will gain a lot of positive charge. What's that going to do to the interior, the potential difference? What that's going to do will be to swap that potential difference and make the interior more negative than the exterior. And that's why we see that charge go from +30 all the way down to past -70, because we are having a mass exodus of positive potassium species from the interior to the exterior. And because of that, we get a change in potential difference. The interior now becomes more negative relative to the exterior. Okay? And that is that repolarizing phase. At +30, sodium channels close, voltage-gated channels open. Why could potassium not move before? Because the gates that allow it to move were closed. But we hit the right voltage for it, about +30 millivolts. And that allowed those gates to open up. And that causes those gates to open up and allows the potassium to flow from the inside to the outside. What does that do to the charge of it, the potential difference of that node? Well, you got positive species leaving. You got positive species leaving. What's that going to do to the interior charge relative to the exterior? It's going to reduce it. It's going to push it into that negative zone once again. Okay. Now, very important, this whole spike up and down in charge, right? Where does this happen? You have to, let me go back to one point here. This spike in charge, where is that going to happen? That whole thing we've talked about, right? It happens along the nodes of Ranvier. Okay, so let's start from the left. Remember we said that spike going up and down, right? Well, that's going to happen at the far left. See that last domino on the upper left? That's going to happen on the far left. We're going to have that spike up and down. And that change in charge will move in the interior of the axon to the next node of Ranvier. And that will cause the same cycle to happen, where the channels open up and allow the sodium and potassium to move. And it goes to the next, and the next, and the next. And each of the consecutive nodes of Ranvier, they will have that spike in charge and then come back down. And that change in charge happens at each of the nodes. And that's how the whole thing works. That we have that action potential, that change in charge at each node, consecutive node, and that passes down the axon over and over. And think of that change in charge as a domino falling. When that domino falls, you got dominoes stacked up, right? When one domino falls, it causes the next domino to fall, and the next domino to fall, and the next domino to fall. Well, here, the change in charge at one node causes a change in charge at the next node, and that change in charge then triggers a change in charge at the next node, and the next node, and the next node, and the next node. And it literally goes like a series of dominoes. Okay. So know that cycle, know what an action potential is, know the forces on an ion. Very important you see the process going on here. Okay, refractory period. During this process, you cannot get a signal to move through the neuron So during this process, we have this idea of refractory period, which is the time that a neuron cannot generate another normal action potential. Okay, think of this as a good example of this. Think of this as the wave. You know, at a stadium, everyone stands up and does the wave, right? People can only do that so fast. Can you get someone standing up and down within a half second? No. Right, or an eighth or a tenth of a second. There is a time when, during this progression of a signal, that another signal cannot overlap. Okay, that concept is refractory period. Now we have absolute refractory period, which not even a strong stimulus will begin another action potential. Okay, simply because that system is involved in the movement of one signal, another signal cannot overlap on top of it. Okay, but that's not a very long time. Okay, we can get approximately 1,000 impulses along a neuron per second. Okay, so that's not a very long time. The relative refractory period is if you have a lot of stimulus, you can bring it back to threshold voltage fairly easily, but to get it back to that threshold of minus 55 takes a lot more stimulus, and that's the relative refractory period. Because we can get it, we need to get it to that minus 55. Okay, we see the dotted line, we see a solid line on the bottom, and then a dotted line going across the bottom, that first dotted line from the bottom, okay, that's minus 55 millivolts. We can bring it back to that point and get another signal to progress, but it's harder to get it to that minus 55 millivolt level, that threshold voltage. Okay. Now, the idea of moving signal, between the different nodes of Ranvier, that idea is called saltatory conduction. Okay. The other one with non-myelinated or unmyelinated fibers is continuous conduction. Okay. Saltatory is going to be faster. Okay, so now we have gotten, this is an important point, is when we get that change in charge to the end of the synapse, the synaptic bulb, what happens? Something has to happen, right? What's going to happen is calcium, that change in charge, will open up a voltage-gated channel for calcium. Okay, and calcium will now come inside the synaptic bulb. And when calcium comes in, it's not shown here, and we're not going to get too in-depth in it for the class, but there's something called snare proteins. This calcium allows these synaptic vesicles to fuse with the exterior or the bottom part of the synaptic bulb, shown with the number 3. When that happens, the balls, the synaptic vesicles, the balls of neurotransmitter fuse with the bottom of the synaptic bulb, and they allow the neurotransmitter to leave through exocytosis. And as that neurotransmitter leaves, it moves to the postsynaptic cell, okay, a muscle, for example. And that will act as a ligand and open channels. You talked about this with muscle physiology in the first semester. Remember, you talked about how muscles are activated and neurotransmitters come down and open up ion channels and things come in and start a muscle to contract. Well, this is how it happens. This is the before of that cycle. Okay. Neurotransmitters open up ligand-gated channels, and you have some action in the postsynaptic neuron or muscle. Now, not always are you going to open up a ligand-gated channel. Many times, this is going to set up something called cyclic AMP or a cyclic AMP pathway. It's going to cause a signaling molecule to form in the postsynaptic cell. I'm not going to worry too much about that at this point, but just be aware that not always is the target a ligand-gated channel. It can be another pathway entirely. Removal of neurotransmitters. Final point here is neurotransmitters need to be removed because they can't keep stimulating. If you have constant stimulation, you're overstimulating that nerve. Okay, you have to get rid of that neurotransmitter once it's done its job. Couple ways: diffusion, right? Concentrated. It's more concentrated in the cleft, so it's going to just by chemical diffusion move out from that. Sometimes you have to use enzymes to degrade it, and then other times you're going to reuptake that by neurons or glial cells surrounding it. And we talked about neurotransmitters later on. We talked about reuptake inhibitors. Sometimes you want to prevent that or delay that reuptake because that's the basis of a lot of mood-altering drugs, Prozac, for example.